In this lecture, I want to go through some examples of reactions that I caught in chapter 1 that I thought were really interesting. So the first one is kind of Lewis acids in biology. Or biological systems. And, you know, we've messed around with Lewis acids before. We say these are lone pair acceptors. But an example that was shown was a really important example. It's called the iron sulfur cluster. So it's iron atoms, and they have 4 sulfur ligands. And we actually get like a box with 4 iron atoms. So I've got an iron atom here, and there we go. Okay, so there's 1, 2, 3 back here is a 4th iron atom, and all of the irons are attached to sulfur atoms in this cluster. Most of the irons are attached to cysteines as their 4th thing. So cysteine is an amino acid, I should say. Complicated picture, and I should add this is somewhere in chapter 1. This is the iron sulfur complex. I'm seeing now that I'm probably not helping making this structure look less overwhelming, but one of these irons, there's an OH2. It's a water ligand. And so what I want to do is just truncate this structure a bit and show S, Fe, S, S, and then we have this OH2. And it's this site that turns out to be reactive as a Lewis acid. And you see these clusters appear in enzymes to accomplish some interesting chemistry. And so what I want to show you is an important reaction where we take citrate, which is the, it's just kind of the biological conjugate base of citric acid. So I'm just going to write truncated over here, just so we see that that's the abbreviated structure. So let's take citrate, which is citric acid minus 3 H pluses. Okay, so citrate looks like this. We've got a CO2 minus because we've lost a hydrogen from a carboxylic acid. We've got a CO2 minus, got a CO2 minus. Kind of looks like glycerol except with carboxylic acids. And then in the central position, we've got an OH. And it's this OH that actually adds to the iron of our iron sulfur complex at that OH position. And so the iron, recall, is attached to 4 things, 3 of which are sulfurs, 1 of which is a water. Okay. And now at this point, we form, after we have that Lewis basic oxygen add to the iron atom, we get to a complex that can undergo an elimination reaction. So we have OH. So I'm not really organizing my structure that well, and that needs to be a better bond. Okay. Now at this point, we've got an OH plus. Now what happens is that in this enzyme active site, and there's actually cool movies of this enzyme active site working, is that you deprotonate at this position, which is alpha to a carboxylic acid and also alpha to our OH plus. You deprotonate here to form and then the electrons of that carbon-hydrogen bond go to form a carbon-carbon double bond, and then that pushes electrons into the oxygen atom, and the iron atom ends up dissociating the water ligand simultaneously. Now what that gives rise to is this product plus an iron sulfur complex, or iron sulfur cluster, excuse me, iron sulfur cluster, which can be turned over by kind of doing some proton exchange. And it looks like perhaps an oxidation state or yeah, anyway, the iron sulfur cluster is regenerated, but we also get a molecule called aconitate. Aconitate, which is an important biological intermediate. Now they call it cis aconitate, which is us just saying, which is a bit lazy and incorrect. If you look at the alkene, it's not cis, it's actually Z, is what they mean to say, right? Cis requires 2 hydrogens on the carbon-carbon with double bond. Here we have 2 priority 1s on the same side, but we don't have hydrogens on the opposite side. We have that other ethyl CO2 minus that soaks up one of the sites. So this should be Z-aconitate, but that's not what they call it. They call it cis-aconitate. Okay. So this is a Lewis acid catalyzed elimination. Now, again, maybe I didn't do a great job of simplifying the iron sulfur complex, but the iron sulfur cluster, iron sulfur cluster is a nice Lewis acid. It can do some other things inside biological systems, but it shows up here and there. And the reaction that it actually accomplishes is just a simple kind of Lewis acid mediated elimination where we strip water off of something. In this case, it's the citric acid to make a new molecule, aconitate. Okay. So let's keep going. Let's do some SN1-like reactions. And so for an SN1-like reaction, what we need is we need to take some sort of tertiary carbon with a good leaving group and let that walk away to form a stable carbocation plus the leaving group as a negative. Now, it turns out that in many cases, the leaving group in biology is a diphosphate. So we kind of abbreviate that as OPP. So that's diphosphate. Now we've already looked at diphosphates before. It's when you've got the O bound to a P, which then alternates OPO until you hit to the end, and then it's an O minus. Double bonds on top of the P, and then single bond to an O minus down below. So this is our diphosphate leaving group. This collectively is a good leaving group. Turns out that when we use ATP, when we actually consume ATP, we push off the diphosphate leaving group to kind of release some energy, which allows us to push some reactions forward. And we'll get to that in a lecture or so. Okay, just a second. I need to take this text. I need to pause the video. Sorry, super unprofessional. Okay, and we're back. Sorry about that. Sorry about that. Here we go. Okay, so back in front of us. So we have this good leaving group. It's used in ATP. It's used in many cases for biological systems. We abbreviate it as OPP, just 2 P's for 2 phosphates. Okay, so let's do some chemistry with it. I'm going to take this molecule and put an OPP here along with just a methyl for added measure. Okay, now what I want to do is I want to let the OPP leave because it's a good leaving group. When it leaves, it's going to generate a carbocation. This carbocation is especially stable because A, it's tertiary, and B, it's allylic. And allylic carbocations are especially stable. Okay, because we can draw resonance structures to them. Not the greatest resonance structure, but it helps us see what's going to happen next. If I push the electrons up to make a carbon-carbon double bond going up and to the right, I'm going to leave behind a positive charge at the less substituted primary position, but we're still allylic, so it's okay. We can handle allylic carbocations in the primary position. Now at this point, from this resonance structure, it might be a little bit more clear to see that this pi bond can reach up and attack over here. That's going to assemble a ring that's 1, 2, 3, 4, 5, 6 atoms big, which gives rise to this structure. Okay, now this structure can do a couple of things. We can eliminate hydrogen from this structure, or we can accept an alcohol from water. Let's go ahead and do that. I'm going to let neutral water attack the carbocation. Sorry, forgot I needed to make a page. And then after that attacks, we're going to form a protonated alcohol, which is going to lose a hydrogen cation to give an alcohol product. This, I didn't make note of this structure. This looks a lot like menthol. Maybe I'm wrong about that. Maybe menthol has, definitely have that isopropyl group hanging off, but maybe we've got the alcohol elsewhere in the molecule. But anyway, we get to interesting structures. So this is an example of a carbocation rearrangement that gives rise to a new ring. And this is fairly common in lipid chemistry, in particular polyisoprene chemistry. So these are common to a class of molecules called terpenes, and we'll get to those later. Terpenes are polymers, or oligomers, I should say, of isoprene, which is this. So this molecule over here was constructed by a few of these molecules with some rearrangements that occurred. And recall, we have that carbocation rearrangement that gave rise. So it looks like we have maybe 3 isoprenes coming together for this molecule with some other things happening. Anyway, okay. Let's get back to the chemistry here. So we can do, if that's an SN1 reaction, we can do our old favorite, the SN2 reaction. So ATP turns out uses an SN2-like reaction with the diphosphate being a good leaving group, but we can also use SAM. So SAM is S-adenosine, I think it's S-adenosine. Let's go adenosine. Yeah, it's definitely zinil. Methionine. Okay, so it's a big molecule, but there's a few focal points. First of all, we have our methionine, which I'm going to draw as the conjugate base. So there's my amino acid. Now we're going to go 2 carbons away to an S group, which is unlike cysteine, and we're going to have a CH3 sticking off. So there's my methionine. Now it's S-adenosinil because we put an adenosine group onto the methionine. Now, adenosine is a combination of a sugar and a base, an alkaloid base. It's not a bad structure to memorize, but I haven't memorized it yet. Okay. So this whole thing together is adenine. And we put the adenine group onto the sulfur atom of methionine, giving us a nice whopping positive charge on the sulfur atom. Now, what that basically means is we've got a CH3 attached to a positively charged sulfur atom, okay, which I want you to think of as like a really souped up version of methyl iodide or methyl halide. That is, we have a great leaving group attached to a methyl group. So this is like methyl iodide or methyl bromide. So we can do SN2 reactions on it, which is pretty cool. So for example, we can take the molecule shown here, which has an OH and NH and a catechol-like structure, and this is norepinephrine. Norepinephrine, and with this will react with SAM at the nitrogen atom through an SN2-like reaction to give us N-methyl epinephrine or epinephrine. It's probably just epinephrine, right? It's not, because there's the methyl group is always at the nitrogen. So I'm going to write this CH3 here. And we'll lose H plus along the way because this is facilitated by an enzyme. I don't want to get over curly arrow. That's not supposed to be a wedge. That's just supposed to be a not disgusting line to my nitrogen atom. Okay. So we have an OH here and an OH here. And so we went from norepinephrine to epinephrine. Here's a quick hint for nomenclature. Anytime you hear nor, this is the demethylated version of another molecule. Now this is an important reaction because epinephrine, this is adrenaline. So you put this, when you release adrenaline, you're releasing this to be a, what's the, it's a special type of neurotransmitter that can, like when you release, usually when you have neurotransmitters swimming around, they just like sort of act like transistors in a computer, much more complicated than that, but like, 0, 1 here and there, send a few signals. And then this is, escaping with the term is this is one of these neurotransmitters that communicates a broad signal to the brain, allowing you to like drop everything and start doing your fight or flight response immediately. And so like all of a sudden you're not hungry anymore. You say the adrenaline's kicking in because the adrenaline's stopping you from doing other things. Okay. Anyway, so that's a reaction. It's an SN2 like reaction. It turns out, let's see, I was looking at this reaction. It's a complicated reaction. I can at least write it. So this is another sort of cationic reaction I wanted to mention now, but you can do other substitution-like reactions where if you take phenol, you can, or excuse me, tyrosine, the phenol of tyrosine, which is an amino acid, and you react that with the enzyme thyroid peroxidase, I think is what my notes say. This does the net kind of conversion of I minus into an equivalent of I plus. I'm going to put that in quotes. So what you get is an electrophilic iodine, which allows you to put iodine onto electron-rich aromatic rings through like an electrophilic aromatic substitution reaction. And you get to a structure like this. And if you do this right, you can get to the hormone thyroxine. So this is, I believe, an intermediate to thyroxine. I guess that's kind of a weirder reaction, but this is something that happens in the thyroid. So if you've known anyone that's had thyroid cancer, afterwards they have to have a hormone supplement for thyroxine, I believe, because their thyroid's generally removed. Additionally, a nice way to track the to see what's going on in the thyroid for imaging purposes is to use radioactive iodine because it localizes itself into the thyroid. So it's easy to kind of tell. And that's where our body does all of its iodine chemistry is in the thyroid. And it's for this reason, for making this hormone. And so you can really track to see what's kind of going on in terms of if there's some sort of tissue that you need to watch and that sort of thing. Okay. So those are some interesting kind of substitution reactions I thought that were worth looking at. We can look at some reactions involving carbonyls now. And these are definitely a mainstay of biochemistry. Now, the one I want us to kind of be as aware of, or as kind of knee-jerk response to, as the keto-enol tautomerism, is the formation of a Schiff base. I believe I should have capitalized that. I believe Schiff is a scientist, but or whoever decided to name this. But what this really is this is taking a carbonyl and reacting that carbonyl with some sort of amine to form an imine with loss of H2O. Now, this imine, I guess, is a troublesome word, so instead Schiff decided to start calling these things Schiff bases. So if you're in the biochemistry realm, you really don't use the word imine as often, which is probably kind of sad, but probably kind of necessary because it sounds so much like imine. So instead, we're going to call Instead, we're going to call imines Schiff bases. And I want this to be pretty, I want you to be pretty responsive to this. So and we've seen this in so many mechanisms so far in the course. But as soon as you see a carbonyl and an amine, we want to let those two react, lose water, and form a Schiff base, or just form the imine, where we swap the oxygen for the nitrogen along with the R group that comes along for the ride. Okay, so with that reaction kind of in front of us, let's go ahead and talk about an important reaction for the formation of amino acids. Here, we're going to rely on enzymes that use the cofactor PLP. I believe pyridoxal phosphate is the name of PLP. But anyway, it looks like this. So here's PLP. It's a pyridine ring that seems to always be protonated. Okay, and we have an NH2 at this position of our pyridine, an OH to assist with hydrogen bonding, a methyl group, and then an O phosphate, which I show as an OP. That arrow is kind of making this look a little gross, so I'm just gonna, and I like to circle my phosphates to remind me that it's not a phosphorus atom, but the phosphate functional group. Okay, so this is PLP. This is a cofactor. It's a vitamin that we consume quite a bit, and this allows us to produce amino acids with good configurational control. That is good control of the alpha position. So what we do when what we do with PLP is we take an alpha keto acid. You can see that it's a carboxylic acid, but at the alpha position, we have a ketone. So we say an alpha keto acid, and we react this with our PLP. Now, where does the reaction take place? It takes place on the amine to form a Schiff base imine. And so what happens here is it's the carbonyl that's a ketone, not a carboxylic acid, that forms the Schiff base. So we have our Schiff base with the NH2 of PLP. Got an OH for hydrogen bonding, a CH2 to OP. Okay, there, my best work yet. Okay, now what's pretty cool is this, the heterocycle really sets the molecule up to do a deprotonation after we form the imine. So when we form the imine, what we can do is deprotonate this position next door to the imine. We can push electrons all the way down if you want to see how this looks in terms of resonance stability. When we do that deprotonation, we neutralize the pyridine, but at the expense of aromaticity. So we no longer have aromaticity, but we did do something interesting. Okay, so the pyridine would really like to be aromatic again. But what it can do is it can just use resonance to show that it's still aromatic. So aromaticity still kind of exists. We just have to push electrons through. All right, let's push the electrons here, here, here. Okay, if we do that and we stop there, we've broken the octet on the nitrogen. We don't want to do that because it's still got a lone pair. So we got to continue to push through until we can stop. And there's a really good stopping point at that alpha position of the carboxylic acid. And I notice now I protonated my oxygen. I better just leave that unprotonated for consistency. I'm going to do that here and here. If yours are protonated, you can leave them protonated if you want. I'm going to leave mine negatively charged. Okay. All right, so we now position the negative charge at the alpha position. If we look, though, all we've drawn is a resonance structure, so let's be sure to capture those resonance arrows. But the pyridine is now aromatic again in this resonance depiction, but it's just positively charged. So we have a big negative charge at this alpha position. Well, here's where the enzyme that holds on to our PLP cofactor. It turns out PLP doesn't do its job unless it's inside of an enzyme. The enzyme is going to hand that alpha position a hydrogen atom from one face of the enzyme so that we form the S enantiomer of our alpha amino acid at the end of the day. So the amino acid may seem a little bit far away, but we can at least see the wedge nature coming in from the enzyme. Now, you can't see this from a 2D drawing, but let me just say from enzyme, which means we can control the stereochemistry. So I'm going to redraw this structure kind of. I'm going to abbreviate some of it. I'm going to redraw this structure now with a wedge to the alpha position of the nitrogen because the hydrogen was attached on the back. Now that looks an awful lot like the amino acid alanine. We've put a nitrogen at the alpha position, and now we have the configuration that we get for alanine. And you're thinking to yourself, but it's not an amino acid. It's got this and blah, blah, blah. We've got the rest of our molecule to worry about. But what we also have is a ton of water, and water can be used to do the reverse reaction of the Schiff base formation. Okay, reverse reaction of the Schiff base formation. Which gives an amine plus an aldehyde. Now, it turns out that this is actually PLP. The aldehyde version is PLP. PLP adducts to a nitrogen within the, yeah, let me just stop there. So this is now PLP as the aldehyde. The aldehyde of PLP. But now we have an alpha amino acid. And the enzyme will now do some work to get back to a situation where we can form an imine on another alpha keto acid. And so this is turned over by the enzyme. Okay, so that's kind of how we use Schiff bases productively, at least one example. Schiff bases are sort of everywhere in biochemistry. Okay, what I want to talk about now is alcohol reduction using nicotinamide cofactors. Nicotinamide cofactors. Okay, so with nicotinamide cofactors, what you get is, I believe you use this molecule, which is nicotinamide. I spelled it right before. Looks a lot like nicotine, it turns out, but nicotinamide. What we're going to do is make a cofactor out of it. where you just adduct, I think it's an adenine dinucleotide. Yeah, there's a big molecule here that's based off of adenine. Just gonna look it up. So there's 2 nucleotides. It's just big. So big part of molecule. Okay. And so we would call this NAD, or nicotinamide adenine dinucleotide. And I'm going to go ahead and write this as a little plus to remind myself we're in the plus state. Now, what we can do with nicotinamide is we can let biochemistry add the equivalent of an H minus by way of reduction. So we can do a reduction. And how this is going to work is we're going to break aromaticity fully and neutralize the nitrogen atom, and that's NADH. So you'll see this a lot in your biochemistry textbooks. And what we've done is we've gone from having 1 hydrogen at the northernmost position of our pyridine ring to 1 hydrogen to 2 hydrogens. Now, what's cool about this is one of these 2 hydrogens sort of behave like an H minus in sodium borohydride. So it's a good carbonyl reducing agent. And if you have NADP, it's an NADPH. These are just very similar molecules with phosphates and that sort of thing. FAD is quite a bit different, but it's the same idea. Okay, so what we're going to do here is we are actually going to treat this a lot like sodium borohydride. So before we had sodium borohydride, we'd take a ketone and we'd add sodium borohydride to it, and we'd get an alcohol, where the H is added to the carbonyl, and we follow up with H plus in the workup to help neutralize. That's a reaction we talked about in organic 2. We did a lab on it, that sort of thing. It was fun. Here we're going to do the exact same thing, except biology can't handle sodium borohydride. Instead, it uses NAD and NAD plus. We have R, and we're going to take our NADH, and here's how this works. We're going to take the lone pair of electrons in the pyridine. It's going to recommend getting back to aromaticity. Now, to do that, we have to avoid breaking the octet on the northernmost carbon by shoving one of the hydrogens along with its electrons off of the molecule and pushing that onto the carbonyl, pushing up the electrons to give rise to our same alkoxide intermediate, which can just be protonated by water. And we have 2 hydrogens added to our carbonyl. Now, the outcome of our NADH is that it's going to become NAD plus. So we have to have some way, using some sort of enzyme, to turn that over and turn that back into an NAD molecule so that we can do chemistry again, or we can do reductive oxidation chemistry. So one common way is to actually allow this to be an oxidizing agent with other alcohols. So you have some sort of other alcohol around for NAD plus to oxidize. NAD plus will be an oxidant to make more NADH. So these things can kind of work back and forth. And what I'm saying is if we take like a different alcohol, okay, the NAD plus, in the presence of a particular enzymatic environment, can accept the electrons from an alcohol at the CH. Why would it do that? Well, if we take the electrons and the hydrogen atom from an alcohol, we could push the electrons through the pyridine and neutralize the pyridine's positive charge. Now, when we do that, we put the alcohol now in a precarious position. You see that I took a lone pair to help facilitate pushing the electrons off. But that's going to give rise to a carbonyl after we lose a hydrogen atom, hydrogen cation. Okay. So we put the H back onto the molecule and we go from NAD plus to NADH. Okay, so that's a really important reaction. You see that all the time in biology where they're able to accomplish carbonyl oxidations and reductions using this NAD plus and NADH cycle. Okay, and we can abbreviate the bulk of the molecule. We just have to understand that it's that pyridine ring that allows us to accept an H minus, then give an H minus back. It's kind of like how the PLP, everything was kind of about the pyridine ring being able to kind of temporarily avoid being aromatic, then getting back to aromaticity, and seeing what that actually does to the system at play. Yeah, it's a really fascinating molecule. Okay, I should make another page here. Now let's, before we move away from carbonyl addition reactions, we'll talk about carbohydrates briefly and carbonyl additions. So carbohydrates are kind of gnarly structures, chair structure here. And then I remember glucose, beta glucose anyway, is everything is axial. Now at this last carbon up here, we have to have a CH2 to an OH, but we have OHs everywhere except that CH2OH, an oxygen in the middle of the ring, and then everything's equatorial for beta glucose. Okay, so in this case, what we can do is, with the movement of a hydrogen atom, and it's going to be this hydrogen, that hydrogen is going to jump over to the oxygen that's part of the ring, you can ring open this carbohydrate, and this can happen in equilibrium. And I like to, after it's ring open, I like to kind of keep it as a chair structure so I don't lose track of my stereocenters. But I've taken my chair structure and popped the ring open to make an aldehyde. And what this can do is this can actually go backwards, where the alcohol could attack back in with a hydrogen transfer event occurring. And that could give rise to another ring closed carbohydrate. Both of these structures are considered to be the monosaccharide glucose. It just so happens that they exist in equilibrium along with the formation of a product where instead of having the alcohol where the OH is highlighted in red, instead of having that pointed up or equatorial, we could have it pointed down or axial. And the only reason why that one can move either equatorial or axial is because of the equilibrium with the open form aldehyde. Okay. Now this is actually called beta glucose, excuse me, alpha glucose. Let's mix that up. Axial is alpha. Okay, alpha glucose, because now the carbon at this position is pointing downward. And we'll talk about carbohydrates in a future lecture, and we'll think about this position. This is actually called the anomeric position, where you can flip between axial and equatorial. Okay, we have some more reactions to cover, but I feel like I'm getting a little long-winded. So I'm going to cut here, and we'll pick up with conjugate addition reactions in the next lecture. Okay, so until then, I'll see you next time.